Okay. Better? All right. We're not audiovisual technicians, so you'll have to put up with us one way or the other. Is everyone able to see and to read the font? Because you may want to go ahead and get up closer now. And happy to force people to sit in the front row if they absolutely need to. We've all been teaching classes for a long time, and we may not have brought candy to give everyone to lure you up to the front, but hopefully this will be sufficient. Luther, are you going to give me the thumbs up when you're ready? Ready? All right. Welcome, everybody. My name is Dr. David Brown. I am of the Fairfield Foundation, and I get to the honor to kind of kick us off today. It was my great pleasure to be involved with this project, not only because as an archaeologist, I really enjoy collaborative engagement with communities, but I also have a strange and wonderful connection with this church and the congregation that I didn't know until the day that I stepped foot on the property. I grew up in Pocosin, Virginia. I'm a military brat. I've never really considered myself from anywhere, but in my upbringing, my parents raised me Catholic, and the most meaningful priest who ever guided me in my childhood and helped me understand Catholicism just so happened to have recently been transferred from St. Mary's to the brand new church in Pocosin, Virginia. And so Father Thomas Quinlan was my priest. And he had previously been the priest here. And knowing that the connection between the two, immediately knowing people who knew all of the intricacies of TQ, as we used to call them, both the ups and the downs, meant that I immediately had something in common, and with a very sensitive subject of human remains and working with the complexities of race and the complexities of urban versus redevelopment. I thought that I would be immediately able to talk with people knowing that there was something that we shared. And that made a great deal to me. As an archaeologist, I'm often called into situations like this where archaeology comes to the forefront, and at the same time think about all of the ways that it can add to our understanding of the past, our connectivity with people in the past, and the history often guiding us as we learn from it. So for us, it was just opening so many doors at a time in which so much was being closed. I will talk about it during the presentation, but one of the most influential parts of this experience was the COVID-19 epidemic. And so experiencing that, doing so in a welcoming place that couldn't be as welcoming as it wanted to be, understanding that the limitations brought a lot of history into our fingertips, studying that the yellow fever epidemic of the 1850s was just as influential to the church and its predecessor, St. Patrick's, meant that we were constantly in the feeling of repeating history as we were looking through, excavating from the ground and thinking about what we might learn from the past and how it might help us today. So my hope is that that short preamble will set the tone for what we're going to talk about now, which is a lot about the past, but a little bit about how it changes us in the present. For those of you who don't know, when we were first called in on the project, Paige gave me a call at Commonwealth Preservation Group and said, we think we found something. Luckily for us, it's what you see here in the photo. Let's see if I can get the, I'm deathly afraid I'm gonna go ahead and click on the wrong thing. Hey, I got it, nice. So what you see in the photograph here is a tunnel. We're not going to be talking much about the tunnel today. Sadly, that's a lecture for a different day. But the excavation of the floor area between the pews and the front door of the church exposed for the first time since the 1980s a section of drain that everyone was quite fascinated with. They were like, how could this be a burial underneath the church? Because there was a general understanding there were burials nearby. And so I came in, I got to take a look, I looked into the hole. It was very, very long. I immediately knew that the tunnel was a tunnel and not a crypt. And so we were quite happy to hear that. But as we looked around, there were construction teams who were looking at other areas, particularly under where the planned bathrooms were, and they just found voids in the floor where lots of brick had amassed over time, but for some reason showed there was something deeper. So as we peeled back a little bit of the layer at the top, we started finding the outlines for crypts. And we knew that while the drain may have brought us through the door, the crypts were what were going to keep us here and set the basis for what was to come. In this very complicated graphic you see here, I'm going to be focusing very briefly on the area that's right here. And you can see this is area where the floor had to be lowered slightly so as to go ahead and to make it easier for people moving from one end of the church to the other. And you can see quite a few things here with numbers. These are all different burials or crypts that were found beneath the floor. And we understood that most of them would not be further impacted by the church's restoration, that they had been under the church since the 1850s. They could remain under the church. And because people had an understanding of that, they also knew that the cemetery on the side, you certainly had graves there. And eventually, when we did scrape down in front of the church for the new planting beds, we found even more graves there. And so it's just the existence of a Catholic church in an urban area. This is reality, that the layers and layers of history, one on top of the other. What was always welcoming to that was that there was such great interest because they knew, the congregation knew this was St. Patrick's burial ground. And when St. Mary's came about, there was really not an artificial break between those congregations, that there was a continuity, a respect, a desire to understand and to be felt a part of a long-standing community all the way back to the formation of the church here in Norfolk in the 1790s. And so today we're going to talk about several of the burials, particularly ones in Crypt 1, 2, 3, and then the human remains recovered from burials 7 and 8. Now, for a lot of excavations, there are archaeologists who come in and do the work under contract, usually a state permit or a federal permit, funding that's meant to lead the way for roads or for other community improvements. This is not that. And so we knew from the beginning, rather than just assume a process, we needed to make sure a process was in place where the congregation was playing a prominent role in the decision-making from day zero. When we were brought in and identified human remains were there, we needed to engage with the state. The state is a community of bureaucrats and other folks who are helping to protect us through some permits, but also to ensure a high level of quality in the work that we were doing. Beyond that, though, the congregants were the ones who really were setting the stage for what we would be doing. So it was on myself, on Commonwealth Preservation Group, on my colleagues who were experts in the field of archaeogenomics, to go ahead and to talk about the possibilities and to share our understanding with the congregation at every stage. So for us, it was a joy to be able to have meetings with the congregation, even during COVID, where we could share what we knew. And that those moments to be able to say, these are the options that could help if you're interested in learning more about these as we move forward. Not forcing the congregation's hands, but giving them a sense that they were in control, because honestly, none of this would have happened unless the congregation was in support. So for us, sharing, even in the most difficult of conditions, is important, because archaeology is not anything more than very sophisticated trash collection. In this case, it was the most meaningful engagement, when you were literally sharing history and discovery at the fingertips and then throughout so many people's lives, and in honor of my career. So community continues until today. And I get to hand this over to my colleagues now, because discussing what comes from here, we think will be an influential and important chapter in the history of the congregations. And so we can do the quick Q&As, otherwise we can save it till the end. So this one here, this is actually crypt number 1. And crypt number 1 is directly underneath the stained glass window that you see in the back section of the church. No, it's okay. It's easy to get turned around even with a beautiful place like this. Symmetry is an architecture lecture that I'm sure Marcus and Paige could probably handle better than I could. So, but yes, it does definitely look a lot different, doesn't it? And so from here, I'm going to hand it over to Tad. Are you going handheld, or are you going to go with this guy? All right, I'm going to turn this off for fear that it will be reverberating.
So good afternoon, everyone. I'm going to be providing, so Dave's presentation said it very nicely, provided the background and for the project itself, what motivated it, what the questions that will emerge from talking about recovering human remains and studying their ancestry, and the history that's linked to those burials and who those persons were. So my part of the presentation is to provide as much as we can through archival research and other information we can gather, historically speaking, what is the context for the emergence of the Catholic Church here, St. Patrick's, the building of St. Mary's, connections between the communities, which may have some influence on how we interpret what we find when we start to do the genetic research. And the archaeogenetic research will be described in some detail by Dr. Raquel Flescas. So, the historical context. So, the main goal, at least from a DNA standpoint, is to trace the ancestry of the decedents whose remains were disinterred and studied through the methods that Dr. Flescas will describe. And then situating them in this kind of a history within St. Patrick's, the emergence of the Catholic community, the formation of the actually immigration into Norfolk that actually led to these communities emerging. So what I'll present now are various lines of evidence which suggest who these persons might be, where they might have come from based on what we know about the history of Catholicism very broadly, and also Catholicism here in Norfolk. And I speak not as an expert in this area, but as someone who's very much interested party, who does anthropological research, archival research, as well as genetic research, and tries to do this kind of synthesis of information to understand the DNA data that we generate from different populations, both living and also the past. So Catholicism in what's now the Americas began, as far as we can see, at least in a formal way, with the formation of the state of Maryland, or the colony of Maryland, named after Henry Maria, the wife of King Charles I, whose images you see here in this particular set of paintings. The Maryland Tolerance Act was also established at that point, allowing for both Catholics to settle there, but also welcomed other worshipers to this area, including Anglicans. But sometime later, a few decades later, things changed. With the glorious revolution of 1688 in England, Protestants assumed control of the throne of England, supplanting King James, as you'll see, and then the diminished Catholic authority in Maryland itself. And then you see right here that King James was deposed at this point and replaced by Mary II and her husband, who was actually a nephew of James. So this transformation or change in political power, and the change in the political winds, influenced where Catholics decided to live. And one place they came actually was Norfolk, as far as we can tell from the reconstruction. Probably some places like Spain and England, some from Maryland, of course, may also have come from France and Haiti in the wake of revolutions there in the early part of the 19th century. And so these kinds of details suggest where these persons might have come from, and hence what their genetic ancestry might be when we do the DNA analysis. But we also, so these are just to illustrate these points. The Haitian Revolution actually was a transformative event in world history. The first time that freed blacks and African Americans overthrew an empire, the French, although it went back and forth with powers afterwards. But this was really a fledgling moment in terms of burgeoning democracies around the world. But it also led to some amount of instability, which led persons perhaps to come to the Americas, to Louisiana, maybe to other parts of the South. We also know that this time period here, just as St. Patrick's was being built, was a period of the Irish potato famine, and led to a huge influx of persons of Irish descent to come to this country. documented in various kinds of ways. So persons here might have been of Irish descent. We'd also know that over time, if we look at this time period, especially the earlier part, almost 50 million people came to the United States from various places, including places where you have strong Catholic presence and populations. Italy, to some extent, Ireland, and other places of this sort. Interesting, France in this time contributed relatively little in terms of the population, although there may be, well, as we'll see, that's something that we'll see some results which belie some of these statistics. But in any case, by surveying this, we have some idea also in a general sense where people might have come from to various parts of the United States, including Norfolk and Virginia and places close to it. So just in terms of a religious map, this is of the late 19th century, but this is probably relatively accurate for mid to early 19th century. You can see the various places where you have a majority Catholic rule or Catholic presence, where in fact immigrants might have come to the Americas and settled with communities that were familiar to them and who shared similar values in terms of worship. So this changing landscape also is occurring at the same time. I mean, in terms of communities, they've mentioned the continuity of communities between St. Patrick's and St. Mary's and all the forces which influenced this process. And so in this case, other migrators who came to this part of the world, of course, came involuntarily. And so we know, in fact, that even though there was an outlaw of the transatlantic slave trade, Norfolk was involved in actually the internal slave trade with regard to African Americans is based on historical information we have here in Charleston and other places, including the connection between New Orleans and Norfolk in this regard. So the historical documentation of this, there's I think a couple of projects now being published. Evidence of this in terms of ship logs and so forth, it's the same thing we see when we're doing work in Charleston with looking at the history of African American communities there from the 18th century. And various records show that, in fact, that a number of skilled artisans, emerging culture, and so forth emerged within the 18th century, also during the time that Catholics were coming to this area of the world, although there were pushback against the emergent process of population growth and with these various kinds of sort of punitive and restrictive laws. But in fact, by the time we get to the 19th century, before the church was built, African Americans made up over 40% of the city population at that point. And you have various areas this may be familiar to those of you who are from this area, but a large population in downtown and various places within churches where there's spaces for communities to arise and to worship together. So we also know, based on history, and thanks also to Paige Pollard for some of the work she's done on all of this, that most communities, most areas of worship were not segregated. But in fact, by this time in 1824, the Catholic priest Christopher Delaney, actually had somewhat of an integrated congregation. And these masses actually were continued, including new immigrants from Europe and from other places. This, of course, led to part of the story, which is how St. Patrick's wound up being destroyed, being burned by, at least supposedly, by the Know Nothing Party, who are anti-immigrant and racist in terms of their perspectives and didn't like mixed congregation worshiping. So this burning of the church, of course, then led to the effort to rebuild the Catholic community and the creation of St. Mary's in 1858, including, this is being rebuilt, as Dave says, on top of, most likely, the St. Patrick's Cemetery. We only have looked at part of it through the excavations. We have other evidence all around the building as well. So this was then, this is history I'm sure you're familiar with as members of the church, but this is now a significance in all sorts of ways because of its beauty, its uniqueness, and its consecration as a basilica, one of the few actually, I think, in the United States at all. So this is quite remarkable, but also at the same time, the legacy of this is that it wound up separating communities at least for some time. St. Joseph was the church that emerged in the 1960s, and it converted a number of folks immediately and served the community in this area for quite some time, also opening a school, the first Catholic schools in Norfolk in the late 19th century. But like other things around in urban environments, various kinds of renovations ultimately led to its demolishment for renovation projects. And as a consequence of this, as you well know, the communities then came together here in St. Mary's And now this church is one which has a predominantly African-American community. In this basilica here, this is a photograph actually from the church's Facebook page. So that's the context for the next. So the summary then, just to lead into the DNA evidence, is that we don't know exactly where people would have come from to become members of St. Patrick's. They could come from a number of places, based on what we can reconstruct about Catholic parishioners and worshipers coming both within the United States, other parts of the United States, to Maryland, other immigrants coming here as well, maybe even the Caribbean. So this means that we may see persons with the data we have from these individuals who are disinterred may have different genetic backgrounds for this reason. Again, the separation of the communities for nearly a century, but now predominantly African American, and persons for the current community members may or may not share the same identities as persons who were buried in the 19th century. But again, this is evidence will help us to understand this population history in the 19th century and to help us understand this larger history that I'm describing here. And we'll come back at the end to talk about some of the broader implications that these data together might mean. So with that, I'll conclude this part, and I'll turn things over to Dr. Fleskus. Oh, I'm sorry. I'm sorry.
All right, can you hear me okay? I get to sneak in really quick with a more detailed summary of the archaeology. Now, I told you I wasn't going to talk about the drain much. I do like to highlight, though, for those who weren't around for the initial presentations that we gave, the drain. You can see archaeologist Mike Clem with the Department of Historic Resources. He's actually down in the drain, and we're looking through for any sign that people would have used this as a tunnel to go ahead and to escape, or to go ahead and to do anything other than draining waste or water that was accumulating in urban streets away from the center of town. As all of you know, Water Street is not far from here, and this was essentially one way of keeping the area of Norfolk, this area of Norfolk, from having water basically standing in the streets. The fascinating part about the drain to us is that it was cutting through several of the burials, meaning that it was constructed after this was already an active cemetery. And then even more interesting was that it didn't mark the end of the cemetery's existence. There were additional burials that were cutting through or impacting the actual tunnel itself. And so we know that sometime in the mid-1820s, 1830s is my guess, is that you essentially have this tunnel's creation. We don't have an exact date, we don't have court records, we don't have newspaper records that mention when the tunnel was created, but what we were able to find is that there were multiple tunnels. So even where I'm standing right now, we're over top of a second tunnel, both of which intersect and actually exit the church underneath one of your windows, coinciding with one of the window wells that you have on the outside of the church. We also know this because several animals over time have gotten into this tunnel and left presents for us. Small pieces of trash, or occasionally passed away in the tunnel. So interesting nonetheless. The focus when we were doing the excavations was to very slowly and methodically, documenting each step in the way, remove the disturbed soil that was above the human remains as carefully as possible. In some cases, like with crypt number 1, the top of the vault had collapsed in. And so we were dealing with removing a lot of the machine-made brick that was on the top of it. Machine-made brick comes in at the beginning of the 19th century, and it becomes more and more popular as you get close to the midpoint of the century. So it was not a surprise for us to find things that look more similar to what you encounter in homes today. And in the wealthier of the tombs, you would often also find things like personal items. We found bone buttons, and we found remains of coffins that were quite sophisticated and quite fancy. And so they had iron handles, and they had hinges. In some cases, they also had other details to them that kind of set them apart. Much like we do today, you have all range of different ways of interring humans into the ground, and this was one example for us that we then saw in variation across the rest. A quick shout out to Joanna Wilson Green, who is the state's cemetery archaeologist. She's listed here, or photographed here. She also manages the African American Cemeteries Fund for the state of Virginia, so for congregants interested in helping receive some support from the state for the maintenance of cemeteries, she would be the lady that you would reach out to. I always try to promote this program as it was passed, I think back maybe about 5, 6, 7 years ago, but slowly has become more and more accessible to congregations across the state of Virginia. And so someone you should get to know, because Joanna's a wonderful human being. And this is Keenan Hurdle, who also is a local, lives over closer to Great Bridge, and a longtime volunteer of ours. He was able to come in and participate. He was the only volunteer in the history of the project to go ahead and to work with us in that capacity. What you see here very briefly is the other end of the range of different internments. In this case, you can see our crews excavating down into what is called a lawn crypt. A lawn crypt is where you would bury multiple family members under or within the same chamber. This one was intended to have 5, maybe 6 individuals inside. When we excavated, we realized that it had been exhumed prior to the construction of St. Mary's. And so all we found was an iron bar that would have separated the bottommost coffin from the one that was suspended above it. And in this case, found small bits of human remains that we were able to incorporate into our study. But these were simply the pieces that existed that the church's congregation, those members assigned with moving the burials to the new cemetery, were unable to go ahead and to bring with them. They did a fairly good job of putting the soil back in. There was 6 feet of soil in this, and one of the most difficult excavations our team has ever taken on. I'm going to quickly transition over. I know I didn't have an illustration of it, but we do have dirt burials, if you will. These are burials that would not have included crypts or brick lining. It's important to highlight that these don't necessarily date earlier or later, but definitely exhibit families or individuals who had to make a decision about how to bury their dead, who likely didn't have the economic ability to go ahead and to have a full crypt installed. These could have been occurring in the 1790s, early 1800s, or they could have gone all the way up until the end of the cemetery's use. The important thing to understand in this case is that they may have also been something that was more expedient in burial. You can think about the tremendous amount of labor necessary in order to go ahead and to make a brick crypt. You're not only excavating the hole, but also laying up a foundation around it, almost like constructing a small building. With a burial of a wood coffin in the dirt, it's a much simpler process, one that honestly the rest of us are all much more familiar with than the actual erection of a crypt. And in our instance, we were also curious to find that there were certain intricacies about these. First, the one that we'll be talking about later, burial 7, had a thick layer of lime or possibly mortar, shell-based mortar that was over top of the burial. Whether this was placed there for some belief that it might have kept the spirits or disease encapsulated within the burial that was beneath. There was a lot of thought at the time because we were trying to learn as much as we could about the yellow fever pandemic, but also the misunderstandings of how disease worked and different ways of looking at hygiene in the 19th century. Like most archaeology, there's more than one interpretation. A second one is simply that mortar or lime is associated with the construction of this major basilica above it. And so we're left with quandary as to whether or not it pertains to one period or the other. The one thing we knew for certain, though, is that the heavy clay that was excavated to make room for these burials, when it was pushed back in on top of the wooden coffins, was packed so tightly that it essentially created a form that when the coffin disintegrated, you could still see the shape of the coffin in the ground. Not only that, you could see the grains of wood that still survived impressed within the clay. The first of the burials we found, burial number 7, was found by accident because there was a small hole that opened up one day when we were done excavating a nearby crypt. And so we lowered a camera down into the hole. And this is the one that you'll hear us talk about where it appears to be a child and a small coffin sitting directly on top of an adult coffin beneath it. The only reason we're able to understand that, first and foremost, is that the child's remains were found directly on top of the parents' remains. But also, as we lowered a camera into it, we were able to turn the camera around in a 360-degree view and to observe the walls of the coffin, not just of the adult, but of the child that was above it. That's seeing the light of the flash hit it at different angles and then reading the grain of the wood. Certainly was a moving experience that brought home the fact that these were real testimonies to the 19th century and the treatment of our ancestors in that way. So happy to talk more about the archaeology after we've concluded with the presentation, but seems fitting, based on me already having a full presentation on this, to hand it over to my colleague.
Good afternoon, everybody. Good afternoon. It's a real pleasure to be here today. We first came to this church in 2020 when the Basilica was under renovation. So it's incredible to see what a beautiful job has been done. And it's really been truly an honor to be involved in this project. And it's really exciting to be able to come back and share with you all results today. So I am going to introduce Dr. Dana Coleman, who unfortunately cannot be here today in person. She is traveling, but she sent us an audio recording from her study on the osteology, or the bones of these ancestors that were interred here at the Basilica. So hopefully, if we can get technology to work today, we'll see if we can get this to play.
Hello everyone, my name is Dana Coleman, and I am the one that did the bioarch.
All right, so what I might do instead, if I could take the liberty, so Dr. Dana Coleman is a forensic anthropologist. She works at Towson University in Baltimore. Okay, she's done a lot of really amazing work, basically studying human skeletal remains in different historic contexts. I've worked with her in some 17th and 18th century contexts. So she was the one who basically did an inventory of the human skeletal remains to assess things like biological sex, right? Men, women, age of the human ancestors, as well as ancestry, okay? So she gave a 3-minute overview. I did listen to it, so I'm gonna try to relay as much as I can what she found. So she took an inventory of those crypts that Dr. Brown talked about earlier on, right? So we were able to take, there were skeletal remains present in burial 1, right? So crypt 1, so that's over by that window there. Number 3, right? So over there in that corner, as well as burial 7, right? So these are the ones that adult and child that Dr. Brown was talking about, as well as in, I think, number 5 as well. Or number 2, right? So right here, that very deep one. So the first step that Dr. Coleman typically does is she'll do an inventory, right? So count how many different bits of the skeletal individual is present. A lot of these individuals were not in really great shape, right? So they had poor skeletal preservation, and that's because sometimes when we are going from dust to dust, right, when we are interred in the ground, we will go back into the ground. So at this time, we kind of caught them in the middle of this degradation process. A number of the individuals were assessed as being likely female. She gives a possible or probable designation because sometimes it can be difficult to tell. So we can look at things like the cranium, right? So the shape of our face, if it's a little bit more gracile or smaller, can tend to indicate a female. We can also look at the shape of the hips, right? Females tend to have a little bit wider hips, so we can look at that. But if we don't have a lot of good preservation, it can make this assessment a little bit more difficult to do. So she determined one individual's male, a lot of possible females, some being indeterminate. This individual here, indeterminate because this individual was 3 to 6 months old, so that's that child right there, right? So too young to determine what sex this individual was. She then assessed age. We can look at things like the wear on bones, right? As we get older too, we have our cranium, right? And the sutures between our different cranial plates will fuse together. So when we get very old, it almost will appear, these suture lines will almost disappear. So these individuals, a little bit older in age, but mostly adult, okay? And ancestry, what Dr. Coleman did was look at different features that may indicate population affinity, right? So some features in our face might look a little bit different depending on if you're European descent or of African descent, or sometimes we can look at the shape of the teeth. So she looked at kind of the molars, the cusps of the molars on the teeth, which may indicate some folks were of African descent. However, I will caution too that the preservation was very poor, so the amount that we can, she can only make these decisions with the information that she has available. So going into this, what we've known so far, right, is that we have a series of burials, right? They're dating likely to St. Patrick's congregation, right? So this is pre-1856, right? And then St. Mary's is built on top of these burials, okay? And that these burials are of adults, right? Mostly, potentially a lot of women, older adults. We do have that one child, and possibly of African descent, although maybe possibly indeterminate, right? Based on skeletal preservation. Does that make sense for everybody? Okay, great. Okay, so what we're gonna do now is we're gonna talk about the DNA research that's been done on this. And we're gonna get into the science, so I would love if you guys have questions as we go along, please raise your hands. I want to make sure that you guys understand this as much as possible, okay? So what is ancient DNA? Well, first let's go through. So ancient DNA is basically, we're gonna be extracting the DNA from these burials in order to understand things like genetic sex, right? Ancestry, where folks come from, and then also if there's any genetic relatedness between the burials. So it's a way of presenting and trying to learn as much as possible from the remains that are found at St. Mary's. I did my PhD at the University of Pennsylvania with Dr. Schwer right here, who was my dissertation advisor at the time. So coming onto the site began in 2019, 2020. We came into the project during the burial excavation. So my work focuses on doing community-based DNA studies on the historic period in the United States. So I work in Jamestown, Virginia, Williamsburg, Virginia. I'm from Maryland myself, also Roman Catholic. My Catholic school upbringing would be very happy right now to be doing a project like this. But I'm really interested in how we think about the history of the United States using this DNA perspective. So, all right, so we were brought into the project in 2020. We collected DNA samples during the excavation of these burials. So it's really critical when we're doing this work, as we'll talk about. DNA from burials is often very fragmented and found in low concentrations. So if we're able to collect the DNA samples from these remains at the time they're being excavated, we can reduce contamination. So we were able to implement those measures, which are very new for the field, during the excavations. We gave a community presentation back in 2022 where Dr. Coleman mostly presented the findings from the osteological results, but also the preliminary archaeological findings. We talked a little bit about what we plan to do for the DNA testing. Lab analyses took place in October of 2022, and I spent the last year doing a lot of computational analyses to be able to bring these results to you all here. So it's always really special when we can kind of bring a project from the start to the end. And this is a picture here of what one of those DNA samples looks like after the DNA has been extracted, right? So this little bone powder at the bottom, and then this liquid here, this dark brown, is where the DNA is located, okay? And I'm probably talking a little bit too fast, so I'm gonna speed it up. Okay, so the research questions that we can answer with DNA, and also from talking with folks early on, people were interested in understanding, is what is the ancestry or genetic similarity of the archaeological individuals? We want to know, are they of African descent or are they of European descent? What is the chromosomal sex of those individuals? Do we see a Y chromosome there, or is it both only X chromosomes? That would indicate a female, right? And are any of these individuals related to each other? So can we compare the DNAs from each of these burials to see, let's say, if we have a mother and a child, or if there are two brothers at play? Okay, so how we can study this is by looking at our DNA, right, and how specifically our DNA is inherited. We have our 3.2 billion base pair whole genome, right? So this is like if you sent your DNA to 23andMe and they analyze your DNA for what areas of the world maybe your ancestors have come from, right? They're looking at this whole genome, your total ancestry. Your total ancestry is a byproduct of all of your ancestors going back about 16 generations. So your DNA is a little bit of your mom's, right, and your biological father's DNA mixed up. And so what we can do is understand then where these DNAs come from when we compare them to the reference panels from across the world. Now, what we also can look at is our mitochondrial DNA. Our mitochondrial DNA is a small plasmid, and it's really special because it's inherited only by your mother. So your mitochondrial DNA is the same as your mother's, and is the same as your grandmother's, and the same as your great-grandmother's. So it's this beautiful line of the maternal history of your family that we can study. And inversely, we have our Y chromosome, right? So if you're a biological male, you have a Y chromosome. We can study that to understand where your father's lines have come from, okay? So we use this kind of DNA inheritance, how our DNA is passed to the next generation, as kind of different lines to look at the past, okay? So now ancient DNA, right? We can study DNA to help us think about where our ancestors came from and how we relate to each other. Well, ancient DNA is DNA extracted from archaeological materials or any organic archaeological materials, right? So that can be human skeletal remains, like in this instance, but we can also get DNA from things like like pottery, right? So I'm doing a project right now where I'm extracting DNA from clay tobacco pipe stems. People used to smoke pipes, right? And their saliva gets deposited in that pipe. We can get DNA from that. We can also get it from things like fecal material, right? We call these coprolites in archaeology language. And then also hair shafts, right? But once we extract that DNA, then we can look at those questions, right, of ancestry, relatedness, and genetic sex. Now, DNA, ancient DNA, is code for a lot of times for just really bad quality DNA. If I got DNA from just you and me today, it would be nice, long fragments that are found in tons of quantity. But unfortunately, ancient DNA is often found in very small amounts, and it's often really degraded, right? Because it's been sitting in the ground for hundreds or thousands of years. And when we also extract that DNA, we also are extracting a lot of other DNA with it. So things like microbial DNA, DNA that's found in the soil or the surrounding environments, right? And also potentially any DNA from myself or anybody else who maybe has touched or interacted with the remains. So basically, this all to be said that the proportion of ancient human DNA is often very small in our total DNA sample, okay? So we have to be really careful to make sure that we're extracting this DNA in a secure and controlled environment to help reduce any contamination, right? So this is the University of Connecticut's Ancient DNA Lab. This is myself. I like to hang out in a little puffy white suit by myself all day, so I'm really excited to be able to talk with you all today, because usually I just am by myself. But we have to wear full personal protective equipment. We got some gloves, we have masks and goggles, right? This also has positive pressure airflow, so it's filtering any air that comes into the room. We have overhead UV lights that will UV the room and decontaminate it overnight. And anybody that comes into this room has to be wearing this full protective equipment to make sure that I'm not extracting DNA from, let's say, myself instead of our burials, right? Because if I extracted DNA from myself, it would be found in much higher proportion and totally overwhelm everything. So when we were in the field during burial excavation, right, so that's this window that's right over there. We're kind of looking towards that back of the church. We were able to implement some of those decontamination measures in the field, right? So being able to this is our special UV crosslinker machine to give that UV light to decontaminate. We got gloves, we have bleach and ethanol. And this allowed us to collect these samples, making sure we can ensure the highest level of sterility moving forward. So we're upping our chances for success. So we were able to take DNA samples from Crypt 1, right, Crypt 2, Burial 7, and Burial 3, okay? We took samples from what is called the petrous temporal region, right? So we have our cranium, and we have our temporal bone that's right here. The petrous bone is kind of the bone that surrounds your inner ear, right? So if you have your ear canal, right, you got a lot of really important things in that ear. We got to protect that. So the bone that surrounds the ear is very, very dense. So the denser the bone, the better the DNA preservation. So we try to target those areas for DNA research, right? So we took petrous bone. We also took metatarsals, which are your foot bones too, based on what was available, okay? All right, so unfortunately, because of time, we can't go through each part of the process of how we go from bone to DNA sample, but basically we go through, it takes about, start to finish, about 2 weeks. So it's a lot of, we have to extract that DNA, we have to prepare it for it to go on a sequencing machine, we got to amplify it, make lots and lots and lots of copies, and then at the very end of that, we're gonna get a lot of what we call DNA sequencing reads. That basically is that lines of A, G, C's, and T's that we all learned in Biology 101, right? And now what we have to do is map it to the human reference genome, right? So we get a bunch, it's kind of like a big puzzle, right? We got all of these fragments of DNA, where do they fit on the human genome? And what we want to do is assess how many times we have reads at different areas of the genome, right? Which basically is what we call coverage, okay? So you see here we have 0.1. What that means is that we have 1x coverage would mean 100% coverage across our 3.2 billion base pair. Here we're covering about 0.1 of that, 0.37 here, and 0.6, right? So this is kind of low, right? If this was a nice modern DNA study, we would be having like 10x coverage, 30x coverage. But because we're ancient DNA and our DNA is very fragmented and small, we tend to have lower coverage. But we have to look at this to make sure, do we have enough of the DNA sequence in order to say something about relatedness and about ancestry and about genetic sex? So for these individuals right here, burial 1, burial 3, and burial 8, we were able to have enough coverage produced. Unfortunately, for burial 2 and then that burial 7, which is that adult and child, we were not able to extract enough DNA from them to say anything with confidence. So unfortunately, we're only going to be able to talk about these 3 individuals here. But as you will find out, we've learned a lot from them, which can help us think about maybe the context for that wider burial ground that's beneath our feet today. Okay. So next, what we got to do, now we know we have DNA, right? Well, we got to ask the question, is it real? Because what can happen is that maybe it's the DNA from us, right? And we got to make sure that it's authentic, okay? So what we do is we assess something called damage patterns. So because this DNA has been sitting in the ground for hundreds of years, it's starting to basically degrade over time. So we look for those patterns of degradation as a clue to say, yes, this is authentic. If it looks really nice and shiny and new, then that's not what we want. We want that to go home, right? So basically this graph is showing us we have these little lips here. Basically these are signs of damage that says, yes, we have authentic DNA. Then we want to make sure that we have low contamination, right? So it's not DNA from myself that's there, but that from the burials itself. So we run these measures of contamination, and you can see here we have nice 95, 97, 96. That means we're good to go. We've kind of passed all of what we can say are quality checks. So we look for this because otherwise we could just be reporting on something erroneous. So the next thing we did was look at relatedness, right? Especially, unfortunately, for that context of those 2 burials here, we weren't able to get the adult and child any good, reliable DNA from them. So I wanted to see, well, what about these other burials? 1, 3, and 8, or excuse me, where's 8 right here? Is there any relatedness between them? Unfortunately, we did not find any relatedness. So these are all unrelated adults, okay? Unrelated adults. And fortunately, we did not get DNA from that adult and child due to low coverage. We can maybe hypothesize that given the burial positioning that potentially they were related, but we will not know for sure, right? Just because it could also be that maybe there's a different kind of kinship. Maybe it's a community kinship that links those adult and child. Maybe it is a mother and a child, or a father and his son, or things like that, but we won't know for sure. So, okay, so we know no relatedness, okay? These are all unrelated adults. Next, what we can do is look at ancestry, right? So where our ancestors are from, okay? And so what we do is we basically will compare the DNAs of these archaeological individuals with a global reference panel from DNAs from persons all across the globe and say, how similar is this DNA to somebody, let's say, living in West Africa or living in Western Europe, okay? So this is called a principal component analysis, and it looks a little bit complicated, but I promise it's not. But what we do is each color represents a population, right? So we have a cluster of green down here, which is East Asia, and every dot represents one person. So you can see all these purple dots are up here. Purple is North Africa, right? So we got all these North African populations, people up here. And we got yellow over here. Yellow is Europe, okay? And what we do is we take our archaeological individual and we say, all right, I'm going to put my DNA and compare it, and however close or where these little black dots, which are the archaeological individuals, fall, is what populations they're most closely related to, okay? And so here we can see, oh, we look like it's a lot of yellow, might be from Europe, but what we want to do is triple check that because we also have some Middle Eastern populations that are close by here too. So this is our first clue, okay? So next what we want to do, because we have in our brain now, okay, these might be people of European descent, but is that the total ancestry, right? If we have mixed ancestry, like maybe 2 generations ago, you're somebody in your family married somebody of Asian descent, that DNA signature will also be present in your genome. So what we do next is run what is called an admixture analysis, which is basically population mixing, okay? We want to see basically how much proportion of that individual's DNA is found with what populations. So again, this looks confusing, but we'll break it down. So we have these kind of big chunks of colors, right? We have this big hunk of green, a little bit of blue for individual 1, big hunk of green and 3, and then 8 has a little bit more of this red. So what we do is we compare these colors, which are what we call ancestry clusters, with worldwide reference populations, okay? And what we see here, I have highlighted in black, is European populations, right? So you can see the same kind of pattern is reflected there, where we have mostly this big green color and a little bit of this red, right? So if we had, let's say, a big chunk of blue, like a much bigger proportion of blue, that might indicate that there was some African ancestry in this individual. But what we see here, because we have mostly this big green, and that's what's seen here in these European populations, right? Likely almost completely of European descent, okay? Does this make sense so far? We're getting into the science real deep. Okay. Okay, so next we can ask, all right, now we know, okay, we got European ancestry, mostly all of European ancestry. Where in Europe might these individuals be coming from? So what we do is we do a little zoom in, okay? So each is a principal component analysis, right? Each dot represents one person. Each little color and shape represents a population. We have our archaeological individuals here in black. You can see we have 8 falling here, 3 and 1. They're falling with populations from France, from the UK, and a little bit from Spain too, right? So this is another indication of, okay, we're looking at northwestern European ancestry, okay? Does that make sense? Yeah, okay, great. All right, so takeaways then for ancestry analysis. Likely these individuals are coming from Northwest European regions. Regions like France, like the UK, maybe Ireland perhaps, or maybe perhaps Spain, okay? So we're gonna keep that in our brains. We're gonna come back to it. So next what we can do is look at that genetic sex, right? So we can look at those clues from our bones to say, well, we might thought we have some people who were female, right, based on the hips and based on the cranium. But we can also look and see, we got that Y chromosome DNA. I mean, is that Y chromosome DNA there or is it not there? And that can give us a clue. So we did an analysis to look at the proportion of X and Y chromosomes, right? If you have Y chromosome, then you're gonna be a biological male, right? So what we found here is that 1 was a male, okay, and that matched our osteological estimation. SDM3 was also a male. Now, Dr. Coleman assessed this individual to be a possible female. So sometimes this can happen, right? Because we all exist on a variation, right? Sometimes men have a little bit bigger hips. Sometimes women tend to look a little bit more masculine, right? We all exist on a spectrum, right? So this is not entirely unsurprising given the poor preservation of those skeletal remains. So that's why it's really important that we layer this genetic with the osteology together. And we can either confirm or maybe provide new evidence. Okay, so we have another male, and then 8 here is a female, right? We don't have any X chromosome, so we got 2 Y chromosomes. We don't have any Y chromosomes, excuse me. So what we can do now that we know that we have a Y chromosome present in 2 of those burials, we can say, okay, well, where is that Y chromosome from? What populations might this individual be derived from on that male side? So this is what we call a phylogenetic tree, okay? And right now it's mirroring the mitochondrial DNA, right? So that mother's line. But we use similar notations for that mitochondrial DNA, the mother's line, and the father's line. That's the Y chromosome, okay? So basically, we know that all modern humans originated in Africa, right? The reason why we know that is because African populations have the greatest amount of genetic diversity, most diverse populations on the planet. And from African populations came forth, right, a subsection of that population left, went to the Near East, moved into Europe, went down into Australasia, through Asia, and into the Americas, right? So if you think about that, all populations derive from African populations, right? And so if you look at this big tree, right, so all this red here is African populations, and we have branching that go throughout, right? So we branch into blue, and then we branch into this green color, and then eventually branching into this brown, right? So we use these notations of letters and numbers to talk about that branching, about the shared evolutionary history. Does that make sense? Okay, all right. Okay, so what we can do now is look at that Y chromosome haplogroup, that series of letters and numbers. What we know is that STM1 and STM3, which are my notations, have a R1b distribution, okay? R1b is found in mostly European populations, but also in some African populations and into Asia, okay? It's a pretty wide distribution, but mostly popular in Europe, okay? And specifically, the specific marker at that Y chromosome for individual 1, most commonly found in northwestern Europe, right? So that aligns with our whole genome estimations. And then STM3 kind of more commonly in Central Europe, Eastern Europe, okay? So that might indicate something a little bit broader of ancestry, at least on that male line, okay? Now we can do the same thing with our mitochondrial DNA, right? Our mother's line, right? That mother's and grandmother's and great-grandmother's line. So what we were able to find is that we have a good amount of coverage, right? 54, 112, 114, so that's great. That's a ton of copies, so we can be very sure of our calls. And we were able to see first our STM1 individual has something called an H1b haplogroup. And so this is a frequency map of where H1b is found. So we see here it's found in Europe. The most populous is kind of in Central Europe, okay? So we have a little bit broader regions there. H1 is probably a very common European mitochondrial haplogroup. Next we have STM3, that is a U5A distribution, okay? Actually U5B is my mitochondrial haplogroup, so it's a little bit similar but different. We can see also found throughout Europe, not in a super great proportion, but still pretty present, about 1 to 4% throughout Europe, right? So this makes sense, maybe we have another European individual. Now what's interesting here is STM8 has an L3F individual, has L3F mitochondrial DNA haplogroup. Now L3F is most commonly found in Africa, okay? And some a little bit in Europe, but not very frequent, right? So darker the color here, the higher the frequency. And so this was a little bit unusual, right? Because if we keep in mind all of that autosomal DNA was showing very strongly European ancestry, right? So if we do have an African ancestry signature in that mitochondrial DNA, right, which is your mother's line, we should see that present in that whole genome ancestry, right? But we only see that European signal. So this took me a long time to figure out. I was like, what is going on here? This is very unusual. So I dug deep into the literature to try to figure out what was going on, right? So here's that, right, that European signature, right, from that admixture plot, right? If we were expecting a little bit more African ancestry, we should have a big chunk of blue, but we don't see that. We only see that green. So the question is, what's going on here? What is the ancestral background of this individual 8? Well, it turns out that the lineage, the specific lineage of this one individual traces to a specific population in the Iberian Peninsula in Spain. Somebody about 10 years ago did a full genetic study of Spain, and they found a population in northern Spain, and it turns out that our one individual here, so this is a big phylogenetic tree of only this haplogroup, and each one of these persons is one individual. Our STM8 individuals right here, and it's clustering, right? It shares a lot of closely related branches with those Spanish Iberian L3F types, which is super interesting. So what ends up happening, and which unfortunately I don't have a map here, is that we have, right, Africa is the source of all modern human populations, right? Sub-branch or subpopulation left Africa, right, and came up through the Strait of Gibraltar, right? Lived there for a little bit of time, about 8,000 years ago, right? And we know we have historic movements, during the Moors, right? That could also lead to that presence of an African haplogroup in Spain, okay? So, and let's see if I have a nice summary. In essence, that means that this individual STM8 likely has maybe potentially a Spanish connection, right? Through Spanish Catholics, but also, a deeper connection with Africa via that mitochondrial DNA haplogroup, even though it might be much, much deeper in time, right, than those, than a 18th century connection that is St. Mary's. So quickly to summarize, we were able to extract DNA from 3 of those individuals. All those individuals appeared to have Northwestern European ancestry, right? We have affinity with Spanish, French, and UK populations. All of those places have really strong Roman Catholic connections, right? So it ties the history of St. Mary's, history of Catholic Norfolk, back to Europe in that time period. And then we have that really unique Iberian Peninsula haplogroup for that individual 8. We found 2 males, 1 female, and a no relatedness between any of those individuals. So is any questions about that? Yes? Only that one individual with this African haplogroup, right? But in turn, even though it appears that this is an African lineage, right? Because mostly it's found that specific branch for STM8 for this individual is actually found in Spain, right? So the bigger haplogroup is present in Africa. Mm-hmm. That was a really wonderful question. So basically, she was pointing out, right, that there's a lot of different cultural groups, right? We're using these kind of nation-state boundaries to talk about ancestry, right? What really, genetics does not mirror nation-state boundaries, right? Yes, and there are many, many, many different ethnicities within the African continent. So when we're doing these types of analysis, we're not just looking, I mean, if we look, see if we go back to here.
Test.
If we break this down, right, I broke it down with North African populations, Sub-Saharan Africa, South Africa. Each of these are also multiple different regions. And so we do this analysis, we do sub-analyses to make sure that the ancestry signal that we're seeing is not just looped to say one general Africa. But if we do see any kind of signature of African ancestry more generally, we would pick it up at this kind of broader level, which then would indicate for us to do kind of a more in-depth analysis as to what areas, what ethnic groups within Africa to be more detailed about that breakdown. Does that make sense?
Thank you. My question is, did you actually do that deeper dive? Because a large number of West Central Africans and West Africans were taken to the Iberian Peninsula 3 centuries before they even came to this country. So my question is, when you did that deep dive, what was the result of that? Because I mean, to say Iberian Peninsula is basically, in my mind, as a historian, not saying anything because people were so intermingled in that area and continue to be.
Yeah, and that's a really great point. So we did do that deep dive, right? So if we, and then of course, and apologies, I'm kind of giving a surface level, pretty quick overview on all of this. But so all of these different, each one of these individuals, right, is coming from Spain, from Northern Africa, wherever L3F haplogroup is found, right? And so we can look is that branching pattern, which, and the specific branch that this one STM individual comes from is for that subsect of that specific Anatolian or the Iberian Peninsula signature. But all of these individuals are being sampled from North Africa, from different parts of Spain, from as broad of an area as possible if they contain this L3F haplogroup. So that's how we're able to get a little bit more specific. Usually we cannot get this specific. Usually we try to stay at this kind of broader level, but because you look and use that mitochondrial lineage, that one lineage, and we have these very specific reference populations, it's kind of, it's really actually quite unusual. But no, I completely agree. You have to do that work to make sure that we can actually say this, and you have to be able to, do all the testing with all of the other reference populations. And in the scope of this presentation, we just kind of presented you the short end, but it did take me about 8 months to figure this out because it was quite perplexing. So yeah. Any other kinds of questions? Happy to answer. Okay. And so I think we'll now move to kind of talking about implications with Dr. Schur. I'll move up. Here we go.
Should I mention that? So we're happy to answer questions after the presentation. If you want to email us and such, this is the initial summary of the work we have to date. There are other nuances here which we didn't present, which may allow for perhaps other explanations, but even with 3 individuals, you can see the complexity of trying to piece together the histories of these persons using the evidence, even with the best technology that's available to us today in terms of sequencing genomes. So the overview, I'm going to present just provide a basic overview as sort of a thought, summarize a few points, and then I'd like to make a few acknowledgments for the people who have supported this work. So even though some of these genetic lineages can be traced back thousands of years, in fact, what we've been focusing on is the more approximate history for the past several centuries. Both persons within the context of the 19th century and perhaps earlier as members of St. Patrick's community, and in the broader history of Charlestonian, of Norfolk in terms of Catholic parishioners. We have shown, we think, that the individuals who are buried here are coming from different places within Europe, at least as far as we can tell at this point, possibly Ireland, Spain, and places of that sort. Perhaps then reflecting the immigrant history of Norfolk and Catholics coming here. This actually, even though we have some answers, it opens up new questions about how we actually interpret this within any kind of documentary, historical records and things of this kind. Even, because also the persons where these lineages trace to are with contemporary populations in the areas where they live now, in Ireland and in Spain and France. Who they were, how they recognize themselves ethnically might not be who they are, today, or were back then in the 19th century. So there's still some complexity here that ultimately we hope to be able to describe. This genetic also is, it's important that this is real details that we may not be able to discern based on textual evidence or archival information. In fact, as Dr. Fluskus and Dr. Brown have indicated, it's the synthesis of this information which gives us the power to make any kind of judgment about who these persons were, where they come from, and the sex and so forth, and even the context in which they were buried, possibly economic class, other things of this sort, which also reflect the history of Catholic communities and of Norfolk more generally. And also, this data allows to reflect on the history of Catholic and Norfolk communities, even among congregants who have trace roots to different sorts of places, but share this common spiritual heritage or kinship. So this is really a unique project in the sense that what binds people in terms of the underlying motivation is to understand who belongs to this community over time and where do they come from and how do our communities endure and thrive over this period of several hundred years of sometimes very difficult circumstances and the destruction of the church. So these are at least some of our thoughts about what we see in this study, and we're obviously interested in your opinions and your views of all of this. There'll be a paper written about this. Hopefully we'll get that finished by the beginning of next year, but when it's ready to be published, we'll also consult with you, or at least provide the access to it so you can give us some feedback on it. So I'll leave this open for further conversation with you all here and afterwards. And also, finally, what we can do is acknowledge all the persons who've supported this work. Dr. Deborah Bolnick runs a laboratory in the University of Connecticut where Dr. Fleskus did the work. All of our universities are involved with this. The NSF supported Raquel doing this research. And then Towson is where Dana Coleman comes from. Obviously, the DHR and the Commonwealth Preservation Group are key for this work to be done. Fairfield Foundation, run by Dave and Thane, and I guess the support of Jamestown, who helps underwrite some of your work in this area. Brian Palmer has been really doing a lot of fantastic photographic work and documenting this project. And then, of course, the church itself, your support of community, and Father Jim Curran has also been essential. It wouldn't be possible, actually, to carry out this work without your support and involvement in this whole research process. And we're extremely grateful for that. And it's it's a real pleasure and honor to be able to do this work with you and be able to describe it to you in the way that we can, at least at this point. So thank you for that, and thank you for your attention to our presentations. And I'm sure, again, any of us would be willing to answer questions that you might have about what we've accomplished, at least to this point.
We've all heard about about parents have been reunited with children, et cetera, through DNA. With this mapping here and these three individuals that they have, is there a possibility that someone now or in the future submits their DNA and they may be linked to one of those three?
Well, I'll have Dr. Fleskus answer. I have a few thoughts, but.
Well, I'm really glad that you brought this up, because this was something that we also wanted to talk to you about too. So, in the practice of doing this DNA testing, right, we took DNA samples, the remains, and all DNA samples have been returned to Fairfield Foundation for reburial too. But we also have that question about what happens to the data, right? This genomic data, which could potentially be used to be able to identify descendants, right? And so we want to talk to you all too about the possibility could exist, right? That we could try to identify descendants. 1860 is pretty far back, but we're looking at, what is that, like 6 to 8 generations back, right? The amount of DNA that we have overlapping at that point is small, but it is still there, right? So now technology is getting better and better. Every day technology is changing, right? So this question about the DNA data, right, if it's something that if people are interested in being able to try to find descendants, right, whether that we want to protect that DNA data that is generated from this study by making sure that it's not being downloaded by a family tree DNA site or a 23andMe site is something we want to talk about, or whether or not that is a priority for you guys in a community to be able to find those actual descendants, and what's the best way of trying to go about that, whether that's posting that data publicly online for folks to try to find those descendants, or kind of trying to keep it in-house. So that's just, I wanted to dovetail that because that was a great question to kind of bring up that point. But in terms of if you can actually find descendants, it's the technology is getting better and better in terms of the bioinformatic analyses. The possibility does exist. I don't know if you want to add to that.
Just briefly. So it's also possible that if you're tracing the male lineages on the Y chromosome and tracing the female lineages, we might find people who actually have the same exact types. But because those lineages are fairly old, or in some cases very specific, with that L3F, whatever that is, then anybody who has that is likely to come from that same place. The Y chromosomes, however, may be types that are shared with many, many different men. And because those lineages arose in some number of generations ago, A lot of men may share the same exact types, but not be genealogically related to each other in terms of direct family relationships over some period of time. So they're kindred, but they may be kindred going back farther than the time period we're talking about now within historical Norfolk in the 19th century. So there's some nuances in that regard, but nevertheless, at least on all the DNA coming from your autosomes, there may be a way of making some connections with other persons using this kind of mapping method that Raquel was mentioning here just a moment ago.
In the consideration for connections, today, this parish, well, the African American parishioners came from a predominant, well, it was black parish right down the street. And when they closed that parish and moved us here, most of the European looking people left. So I would think that the chances of having ties with us today would not be as great as one might think, because we were moved here and we weren't here all along. So if you make a connection, you might find the people in the other parishes in the Tidewater area.
And it might be that, those types of genetic connections either can be informative for some folks, or it may not mean anything, right? You guys are connected to this Catholic community right now too, right? And we have these histories. So that's the thought when we came into this project, knowing the history of St. Mary's and the history of St. Joseph being merged into St. Mary's.